In this video, we'll be talking about an important enzyme, that is Taq polymerase, which is used in the polymerase chain reaction. The Taq polymerase gets its name because it is isolated from a bacterium called Thermus equitax. This bacterium carries this enzyme, that is, it is isolated from here, and the name Taq is given because of this. And it is a thermostable enzyme. It is thermostable and can remain active, that means it remains functional between 75 to 90 degree Celsius. It remains functional at this high temperature also. And we have studied that in the PCR technique, the extension process is done at the stage at the temperature of 72 degrees Celsius. That is the ideal temperature. So we need to have an enzyme which can withstand such high temperature. Other polymerase enzyme cannot be utilized for the polymerase chain reaction because as the temperature increases, they become denatured and they become non-functional. For example, the human polymerase enzyme can work ideally at 37 degree Celsius. So this polymerase enzyme actually helps in the addition of the nucleotide bases and is therefore used in PCR technique and PCR technology for rDNA production. That is recombinant DNA production, and it helps in adding the nucleotide in 5' to 3' direction. So it always adds the bases in 5' to 3' direction, just as other polymerase enzyme also do. Now when we talk of the PCR technique, we should also understand what is the PCR actually used for. Why the PCR technique? Why the PCR? Actually, after the amplification, because it is the process of amplification of DNA fragments. So after amplification, the DNA which is being produced by the PCR can be used in many laboratory procedures. So what is the important point? The amplified DNA fragments can be used for laboratory purposes. Also, they can be used in clinical techniques. The PCR technique is also useful in DNA fingerprinting. That is forensic science, we can use it. Then this technique can also be used for the detection of bacteria and viruses. Especially AIDS. And now we have coronavirus also added to it. You must have heard about the PCR test, that is the coronavirus test, because it's a virus. AIDS is also caused by the virus, and also it helps in the detection of genetic disorders. So diagnosis of genetic disorders. PCR technique therefore is a very important technique and is conducted with the help of Taq polymerase.